that is Pamela Weathers. So what to say about Pamela Weathers? So just I will mention that when I started working with Artemisia annua, that it was 11, 12 years ago, I became a real fan of your research, Pamela, because all the different papers that you publish, and I guess what's happened to me happened to too many people also here. So Pamela is a professor of biology and biotechnology in the Worcester Technological University. She has more than 35 years of experience working in Artemisia annua and its phytochemicals, so she knows many, many, many things. And the talk of today, because I think she will have a talk also tomorrow, the talk of today is the phytochemical changes in clonal Artemisia annua grown in the USA. Okay, so thank you very much for coming.
Wow. I am so delighted to be at a conference where everybody knows Artemisia. Usually I'm the only one, maybe two. So thank you very much for organizing this and for inviting me. I'm just thrilled to be here. So what I want to talk to you about today is some of the agricultural work we've done over actually about 10 or 15 years. So, and I have to say, slide. So you all know about Artemisia artemisinin. That's me next to where we've been growing some. So just you get an idea how tall we can grow it. And so none of this should be new to anyone. I hope it's my introductory slide on this. Why don't we have the next slide? There are a lot of diseases that are susceptible to artemisinin and or Artemisia. And the ones that are in blue are ones that we've had a little bit of work that we've studied on. So this is just taken from the literature, what we know. And the only one that's approved is for malaria with artemisinin. We know Artemisia works on that too. Next slide. And there are also chronic diseases here. Inflammation, and we've done work on inflammation and fibrosis. I'll talk about fibrosis later on because that's our new project. But there's evidence on arthritis and diabetes. So this is really, it is a One Health plant, so let's face it. Next slide. So World Health has concerns. And they say artemisinin levels are too low, so you're going to get artemisinin resistance. They have ignored the synergy of the complexity of the materials in the plant, which is unfortunate. So that's where I mentioned synergism. And the fact that Artemisia afra is also potent against malaria and it has no artemisinin in it. So we can't argue about just artemisinin. All right, next slide. So artemisinin varies depending on its cultivation. So that's a big concern because we want to see consistency, right? So yes, you get variability, but is that due to genetic diversity from seed planting? And I'm going to talk about that a little bit. And their claim is that it's too variable in its artemisinin content. And I would argue not if it's clonal or homozygous. We have worked on the clonal varieties, and I will show you evidence today. So that's what this talk will be about, how we propagate clonally. Next slide. So the two approaches, homozygous plants, and that, I am not a geneticist, but people there in the US have done generated self-pollinated plants and shown that artemisinin content seems to be less variable through a couple generations. Because Artemisia annua, when you do seed plantings, you get a lot of variability. And if I'm in the lab and I want to measure artemisinin, I'll see error bars like this. And I want error bars like that, so I can actually ask good scientific questions. So we now are doing only rooted clonal propagation, and I'll show you what we selected from. My students did this. And we wanted to select a relatively high producing plant for artemisinin. We clonally propagate it by rooted cuttings. So we will take the little shoots of the plant from anywhere, and we just snip it off, take the bottom leaves off, stick it in the soil. We use a little rooting hormone, and we get nice roots in a couple weeks. And then they just get transplanted into pots or, for some of the evidence I'm showing you today, in soil, okay? And I'm going to show you successive crops of field-grown plants. We also look at other phytochemicals because they are also very important. They will synergize with artemisinin. We don't know what all of them are. We know some. Many of them are flavonoids. You don't need to know what that is other than that they're very healthy. Okay? And so flavonoids are something that we kind of look at. We don't know all the compounds, but I'll show you some nice pictures that show you how consistent we can make our plants. Next slide. So when we did clone selection, we published now only on one clone called Sam. How did it get that name? It's my students named them Quagmire, Joe, Dan, Peter, Richard, and Sam. And you go, here's deoxyartemisinin, it's a little hard to see close up, and then there's artemisinin in the red, then artemisinin B, and dihydroartemisinin. And so this looks impressive, but it's got the wrong compound that's really high. We don't want that one. So what we want is this one. And as you can see over here, Sam is what we went for because it has lower amounts of this and pretty high artemisinin. Even though this had higher artemisinin, it had too much of this, and we didn't want that. So we didn't take Quagmire. We've been working with Sam, and actually our yields are much higher now as we've propagated along. Just wanted to show you that. Next slide. So that's the one we went for. And then we wanted to see when is the time to harvest a whole plant. And so we looked at vegetative leaves. This is all published data. I can share it with you. You know, if you ask, just ask me for reprints. I'll send anything. Just put the word Artemisia in the subject line of your email, because then I will open it. Leaves at bolting, we looked at the artemisinin content. Leaves at floral budding, we did this about 10 years ago. Leaves at full flower and post-flowering. So we take our leaves usually just vegetative. These are all very close numbers. And we didn't want to have to deal with the hard floral buds that just because they were hard to deal with. So we wanted very consistent material because we want to do experiments. It's different from, oh, I'm making something for therapeutic use as a tea maybe, where maybe I'm not concerned about that as much. But we needed to be able to ask scientific questions. Next slide. So here's the experiment. It's a 9-year experiment. So we live way up here in Massachusetts, all right? And that's where we grew SAM and had isolated SAM. We then transferred it down to a grower who is helping to sponsor this meeting. Down here, it's Atelier Tomanos down here in Florida. We took this clone, Sam. We never let it go to seed. We never let it go to flower, which in plants, that's sex. So we never let our plants have sex. And so they get to make roots off of cuttings down here. The same clonal cultivar, all right? So that's just some average data that you see, but I'm going to show you more. And just to give you an idea, because I know you're from all over the world, Stowe, Massachusetts, where I grow the plants, that's actually my home. It's about Barcelona in latitude, Barcelona in Spain. And then in Florida, it's about equivalent to Marrakesh in Morocco, which I figure most people kind of know where those cities are, but nobody knows where Stowe is. You know where Boston is, though, maybe. So anyway, that's what we did. And you have to remember that latitude on our planet actually affects plants flowering. So here we are in an equatorial region. It's a constant photoperiod, but where I live, first of all, I get a winter, so I can only grow one crop. And the second thing is that I'm at a very different latitude, and so I get these big changes in photoperiod that you don't get down in the equatorial area. So the plants that I am growing up in Massachusetts actually wouldn't grow very well in the equatorial area, but they offer a wonderful opportunity for research. Next slide, please. So that's an example of the plants growing out. I just rotate them around through the ground in my area where I live. That's my backyard. I have a lot of gardens. And they're rooted cuttings of the Sam cultivar. I plant every May. I harvest late August and just before September 1, because at September 1 is when the plants shift and start to move into flowering. And I don't want that. I'm going to show you data from 9 annual harvests. And when I planted it, I put a little compost in. I didn't add any other fertilizer. I watered it as needed. I'd go out and look at it and go like, okay, it needs water. If we had a drought, it needed more water. We would harvest before September 1. We'd dry the plants. The leaves were manually removed, mainly by me, and I would strip them off the hard stems. And then I'd coarsely sieve them to remove those stems, any smaller stems. And then we would just store them dry in the Ziploc bag in the dark. We just got a big room, 25 degrees. And that's how I stored all of the plant samples that were analyzed out of Massachusetts, okay? And we analyzed them all at the same time. So some are 9 years old and some are 1 year old. Keep that in mind, okay? Next slide, please. Now, the material grown in Florida, this is a commercial operation. I don't have any stake in it. We just are helping them grow something that's consistent in the U.S. because there is no consistent Artemisia in the U.S. And so this is what the operation looks like. It's not real big. Remember I told you about the photoperiod? He has to add a little light at night to prevent the plants from flowering. And that's why it's not good for equatorial region. But he can grow crops all year round in Florida because they don't get a winter, not like our winter. And then this is his processing area where they process the dried leaves. And so they do rooted cuttings and they add the light. They have done raised bed, potted and soil cultivation. I believe they're doing all raised bed now, but I think he's talking about maybe shifting to pots. It just might be a little easier. So they've had commercially stable production as of 2022. Next slide. And before I go on, I'm just going to show you. I brought samples if you want to look at. This is, we can open them up, but he encapsulates the dried leaves. This is for human supplement. And he also makes one for pets, dogs and cats. Same material. So we have analytical methods, too, that I'm going to use. One is GC mass spec, which is a very high-tech method. And we quantify artemisinin in that of a methylene chloride extract. And I can send somebody, if you want, this is a hard method to do, so I don't recommend it. If you can do HPLC mass spec, that's better. But anyway, that's what we were stuck with. We also did thin layer chromatography, and that is much easier to do. And I'm going to show you a lot of data from that too. And that's the conditions where we did terpenoids, and we want to visualize them. And then we have flavonoids, those nice healthy compounds, and we want to visualize them. Do we know everything on every spot that shows up? Do we identify it? No, it's very difficult. But we're going to show you what the profiles look like. Next slide, please. So from GC mass spec analysis, this is 9 years, from 2015 to 2023. The artemisinin content and the milligrams per gram of dry weight. The letters after it are just showing you statistical analysis. And so you can see we had a little variation, we had a high producer a couple years here, but we didn't vary a huge amount. And the average was about 10.3 for artemisinin. We also looked at the total flavonoids. This is the aluminum chloride assay. And we saw that this was the overall content. So next slide. Here I've added the Florida information, so 5 crops in Florida, and it's slightly lower in artemisinin, statistically lower, but I suspect if we were able to do 9 crops, it might be very similar, and I'll show you why in a minute. But look at the flavonoid content. It tripled almost. So it's gone up really high, and wow, you were going, that's incredible. Part of the reason is, don't forget, Florida has intense sun. And in plants, flavonoids can act like a sunscreen. So you have higher UV, it's most of the year, and a lot more intense sun. So I suspect that might be it, but I don't know. But I think they're protecting themselves. And you'll see how it changes over time. All right, next slide. Now we get to the pretty pictures. So this is a terpenoid fingerprint. So we run a sample, just for those who've never done this, you put it down here, it's a chromatography sample, and we spot it on there, all equal amounts applied. These are all the Massachusetts ones. And then we let it run, and up here is artemisinin, and here's the standard artemisinin, so you can track that compound. These others are terpenoids. And you can see that we see a very uniform profile for the most part here. And look, in Florida, it looks very similar. That was surprising. We didn't know what we would see. So we're going, oh, that's interesting. So just in general, it's consistent among successive crops for artemisinin, and even the terpenoids seem to be rather consistent within a geographical location. And there's maybe a little slight variation as you went into Florida. And it seems that even despite that change, you know, it's very slight if it's there. It would take a lot of work to say, oh, it's different in this or that. We can't see it from this. But it appears that all the terpenoids and artemisinin remain relatively constant. So a lot of people are focused on artemisinin, so that was something of interest. Now, next slide. The flavonoids are even prettier, I agree. So Massachusetts, again, the same thing. We have no standard here that we're looking at. And you can see the consistency of the flavonoids, but look at how some of them appear to be in greater concentration or less concentration. So that middle green one seems to drop a bit. And then you have these red ones showing up. There's probably traces of them there. When you look at the original thin layer chromatographies, you can actually see the colors, you know. And this one looks like it's maybe a little bit more intense with some showing up. So we're seeing some, but again, it looks pretty consistent crop to crop to crop, especially in Massachusetts. And then in Florida, it looks pretty consistent too. So we felt that that was a really interesting finding. And our goal was this, we had asked the question, is it consistent? Can we grow a consistent crop? Remember, World Health asked that question. They don't think you can do it. If you do it clonally, I think you can. And that was the message we wanted to query and answer, okay? So, next slide. How did it change? So I'm showing you this because these are three standards, artenuin B, artemisinin, deoxyartemisinin, are our TLC. Here's the material that was sent to Florida, but it's our crop. They grew a crop of it down there. They grew another cutting, and a little later on, some others. So these are a couple, you know, they were playing with cultivation methods. And you can see their initial cutting crops looked much like ours. So that was after they had made cuttings from what we gave them. But then you could see it started to shift. And if you look at the next slide, that's the final version of what their commercial product looks like. And so that just gives you an idea. It took a while to get to the point where they were getting a consistent product down there, all right? So it just was shifting as it was adjusting, and they were playing with conditions. So you have to just keep that in mind. So it took about 2.5 years for them to do this. I mean, they're a very tiny company, you know, just a few people, and it takes time for everybody to do this and to grow it out. So the conclusions, this is the first long-term study of a single clonal cultivar of Artemisia annua that was grown in two very different geographical locations. And the artemisinin content seemed to be consistent and varied somewhat among successive harvests in an area and declined maybe by 10% from Massachusetts to Florida, but that's not certain because we couldn't compare the same number of crops. One case it was 9 crops, the other case we only had 5 to deal with. The artemisinin terpenoid TLC profile was relatively constant between both locations, and the flavonoid profile changed dramatically. and so did the total flavonoid content. So all of this, and you can read the rest, I don't have to read it to you. And so you can see that, this is helping us to understand maybe how we should be propagating or growing these plants. They're very easy to propagate by rooted cuttings. Very, very easy. And it might be that that's how we meet some of the needs for getting acceptance of something, of a medicinal crop, such that, we don't keep running into barriers from regulatory agencies. So next slide. Just to thank these people. So that's Atelier Temenos, and NIH sponsored some of the earlier work. Patrick Arsenault was a PhD student, graduated a lot of years ago, and he was helpful in isolating a bunch of clones from seeds that we had growing, seed plants. And Melissa has been in my lab since she was an undergrad. She is now my lab manager and, keeps the undergrads under control when they do experiments. And then James Kishler, who has helped to sponsor the meeting and provided the samples and is growing it in Florida. So I'd be happy to take questions.
As before.
Thank you very much, Pam. Could you kindly go back to the terpenoid Massachusetts TLC, please?
Can you go backwards? I don't have any control over it, so.
Yeah. We'll tell you when to stop.
Yeah, go backwards. There.
Yeah.
Yep. That one? Yeah.
Yes. So it's very interesting because I don't fully agree that terpenoids are quite that stable. If you look at artemisinin, it might, but if you look at more, at less polar material like the gray spots above artemisinin, they seem to be undergoing a slight increase.
Yeah. So
And maybe it's a longer process, but it's interesting to consider that artemisinin might stay stable, but you had an overall boost of other terpenoids, which seems to be kind of increasing when you see 2022 versus 2015.
yeah, and I think this could be also with thin layer. Have you done thin layer chromatography?
Sure.
We could get a little edge effects, and we might have an edge effect there.
I'm speaking of the less polar one, like the top gray dots above artemisinin. They are clearly more abundant in the later years than in the earlier years.
Yeah, and we don't know what those are.
No.
This is where we need a good chemist to go in and
But it's very
look at a lot
interesting.
of these.
Thank you.
Yeah. Yeah, that's your job.
Pamela, yeah. Seems to me that Florida represents more close the endemic areas, where malaria is occur. And then perhaps in Massachusetts it's good to produce and use in endemic areas. It is one conclusion. I don't know if I'm right. And the other question is about the determination, artemisinin determination. You use gas chromatography.
Yeah.
It's
a horrible way to have to do it, but we didn't have good access to anything else.
Correlation with HPLC?
So we used to use, years ago, HPLC. Not HPLC mass spec, but HPLC. And what happens is we were using the Q260 method, and deoxyartemisinin can elute at exactly the same point as artemisinin. And so we stopped using it because we weren't confident that we were actually measuring artemisinin. And so we moved to a mass spec method where we could identify the molecule through its, mass spec signature. And I have wanted to do LC mass spec. We don't have the person power to put it all together. I'm only got a few people in my lab, so it just takes forever. And when you're working with undergrads, so yeah, I wish I would love to do things much more with an HPLC or LC mass spec system. We have it, but we have to work up the method.
Thank you.
Yeah.
Infected people with your clone.
So anything that we have, we haven't done anything with the Florida material, but everything else that we've published on malaria or anything has been with the Massachusetts version of the SAM clone. That's the only clone we've used for, I don't know, since 2010, I guess. Yeah.
Okay.
Sorry, I forgot to ask that very question. Due to the variation of flavonoids that was very striking when you moved your clone to Florida, it's the same clonal origin, it's the same genetics. Did you made even simpler IC50 assays on Plasmodium falciparum on the flavonoid-enriched phenotype versus the stable Massachusetts phenotype?
No, I'll let you guys do that. I don't have the money to do anything with Plasmodium anymore. We have no funding.
Thank you. I'm Peter Fedesh from Tanzania.
Yeah.
I'm dealing with the Anamedi Tanzania. Mostly Anamedi, we are teaching about Artemisia annua, Anamedi, which is a hybrid seed. As I know, Artemisia annua is not growing well in tropical countries. So how can we do to make sure that we are achieving our requirements?
So are you I'm not quite sure the question you're asking me. So you are working with the Anamed A3? Yes. And so if it's a hybrid and you are always producing new seed through the hybridization, or do you propagate clonally?
Of course, we are getting seeds from Anamed International, but we are teaching our people to make cuttings.
Oh, yeah,
Yeah.
from your hybrid, yeah.
Yes.
Yeah, so then you would want to be able to do some tests to see, it's easy to do the flavonoid analysis. I mean, you send me an email, we'll send you the protocols. Total flavonoids is an aluminum chloride method. You need a spectrophotometer. And for the TLC, it's a little practice, but it's not difficult to do. And it's very cheap. And we got the people down in Florida doing it because he's not a scientist, or a chemist. So yeah, they're doing it down there.
Okay, thank you.
Mm-hmm.
Some more questions?
Yeah, I'm a medical doctor from Tanzania. So through the slides we have seen, there are a certain point showing that this Artemisia, it deals with some of infectious disease. And in Africans, we have got a highly infectious disease, which are so terrible. For example, Ebola. Have you come to think about the role of Artemisia in Ebola? Thank you.
I'm not sure I heard you. Have I thought about for what? For
Ebola.
Ebola?
Yes.
I know it has lots of antiviral effects. I don't know if anybody's ever looked at Ebola. It's a very difficult virus to study because you need very high safety standards, conditions. So I doubt anybody has looked at it. We have been trying to get people to do animal studies for COVID, and you need a BSL 3 facility, so it's very difficult. And they were not very successful at trying to do it. It wasn't that it failed, it's just difficult to do it in animals, which is what they want to do. They can do in vitro, but it's animals. Infect an animal and what happens. That's really what you need to do. And that's very expensive and very difficult to do.
Thank you, Professor, for your good and rich presentation. I have just a small question concerning the store of Artemisia. I have seen somewhere you say we must store it in the dark. Why?
Right. So we put the leaves in a Ziploc bag, And then I just store it in a room that is a constant temperature in our laboratory. And we keep the lights off. That's all. And we don't use that room very much. So, you know, it just stores there. So I have, you know, big bags of Artemisia in there. And we will sometimes go in and get Artemisia. I brought samples for somebody, for example. Go and dig it out, take it out. But, yeah, we've had them for years. We use it. I encapsulate my own and use it. Brought it to Tanzania. But that's all we do. And we did publish a number of years ago showing that the flavonoid content also seems to be, the total flavonoid content is rather consistent. The monoterpenes are lost a little bit. They're very volatile. And so you lose those a little bit over time, but not the flavonoid content. And if you wish to see that information, just send me an email with Artemisia in the subject line, and I will send you the article. Happy to do it for anybody.
Any other questions? No. Pamela, I have a last question for you.
Mm-hmm.
So I was surprised about. Okay, no. So I will do the last question. So he's before.
Yeah. Okay. Yeah. There's one more, which is related to the previous speaker. In terms of drying, how did you dry the samples?
So actually very drying a little bit. So sometimes I hang them on my porch in the shade and let them dry initially. And then I take them into the lab in my car, which smells really good afterwards. I take them in my car and put them into this big, it's like a growth chamber, it's just constant temperature. And so they dry really well in there. So they have a very low moisture content because moisture is your enemy. And so you don't want that. And you don't want a lot of light. So, you know, we just keep them in the dark and dry that way. But, you know, like I have last year's crop. I have not processed it yet just because I've been busy. And now I harvested this year's crop, and I have to process that. So they're sitting there, these big stems, where I just take the leaves off and put them in the bags. So it's nothing really fancy, very simple. And no, you know, no special drying equipment. Now, as opposed to in Florida, he's got a dryer because he dries it pretty quick. He's trying to make a product. And it's he has good certification. He has a certified GMP method, and he's even GAP certification, which is good agricultural procedure.
Okay. So last question, I will do it. So I was wondering, because it was very surprising that in the Florida fields, you get almost 3 times higher content of flavonoids.
Yeah.
So have you gone deeper in detail in order to know which flavonoids they are increased mostly?
No, because we just got that information probably December, January this year. And I would have to work with a good phytochemist. I actually have somebody I'm working with at Penn State, and I keep asking him for some analyses, and I haven't gotten them yet, so I don't know. But I would like to know what they are.
Do you have any clue? I mean, just any idea of what we can...
You know, some of the ones that have been reported in Artemisia, I think chrysoplaneton, you know, we have run some sample standards, but on the TLCs. But I really want him to do it where he does, you know, NMR and he does mass spec analysis. He is a good, you know, organic chemist, and that's what you need to validate that spot is indeed this compound, because there could be other spots. Just like I said, deoxyartemisinin runs with artemisinin in that Q260 HPLC method, so that's why we don't use that.
Okay, so we'll know in the future.
Yeah. Right.
Coming soon.
Okay.
Okay, so no more questions? Okay, so thank you very much for the first session of today. Thank you, Pamela.